Next, the clonal selection theory as it applies to B cells. If a person is infected with a virus or exposed to foreign material, his or her blood soon contains a high concentration of antibodies capable of reacting with foreign substance, which is known as an antigen. So most antigens consist of proteins or polysaccharides, but lipids and nucleic acids can also serve in this capability. How can the body produce antibodies that react specifically with an antigen to which the body is exposed? In other words, how does an antigen induce an adaptive immune response? So initially it was thought that antigen somehow instructed the lymphocytes to produce complementary antibodies. It was suggested that an antigen wraps itself around an antibody molecule, molding the antibody into a shape capable of combining with that particular antigen. In this instructive model, the lymphocyte only gains its ability to produce a specific antibody after its initial contact with the antigen. In 1955, Niels Jern, a Danish immunologist, proposed a radically different mechanism. Jern suggested that the body produces small amounts of randomly structured antibodies in the absence of any antigen. So, as a group, these antibodies would be able to combine with any type of antigen to which a person might someday be exposed. So, according to Jern's model, when a person is exposed to an antigen, the antigen combines with a specific antibody, which somehow leads to the subsequent production of that particular antibody molecule. Thus, in Jern's model, the antigen selects those pre-existing antibodies capable of binding to it. In 1957, the concept of antigen selection of antibodies was expanded into a comprehensive model of antibody formation by the Australian immunologist F. Macfarlane Burnett. So, Burnett's clonal selection theory quickly gained widespread acceptance. The clonal selection of T cells is described in the following section. The main features of B cell clonal selection are the following. Each B cell becomes committed to produce one species of antibody. B cells arise from a Each B cell becomes committed to produce one species of antibody. B cells arise from a population of undifferentiated and indistinguishable progenitor cells. As it differentiates, a B cell becomes committed as the result of DNA rearrangements. So each B cell becomes committed to produce one species of antibody. So B cell arise from a population of undifferentiated and indistinguishable progenitor cells. As it differentiates, a B cell becomes committed as the result of DNA rearrangements to producing only one species of antibody molecule. So thousands of different DNA rearrangements are possible. Once again, I'm going to read this because I am a bit confused. So each B cell becomes committed to produce one species of antibody. B cells arise from a population of undifferentiated and indistinguishable progenitor cells. As it differentiates, a B cell becomes committed as the result of DNA rearrangements to produce only one species of antibody molecule. Thousands of different DNA rearrangements are possible so that different B cells produce different antibody molecules. So thus, even though mature B cells appear identical under the microscope, they can be distinguished by the antibodies they produce. Then B cells become committed to antibody formation in the absence of antigen. The basic repertoire of antibody producing cells that a person will possess throughout his or her lifetime is already present within the lymphoid tissues prior to stimulation by an antigen and is independent of the presence of foreign materials. Each B cell displays its particular antibody on its surface with the antigen reactive portion facing outward. As a result, the cell is coated with antigen receptors that can bind specifically with antigens having a complementary structure. Although most lymphoid cells are never required during a person's lifetime, the immune system is primed to respond immediately to any antigen that a person may be exposed to. The presence of cells with different membrane-bound antibodies can be demonstrated. And antibody production follows selection of B cells by antigen. In most cases, the activation of a B cell by antigen requires the involvement of T cell. A few antigens, however, such as the polysaccharides present in bacterial cell walls, activate B cells by themselves. Antigens of this type are described as thymus independent antigens. A few antigens, however, such as the polysaccharide present in bacterial cell walls, activate B cells by themselves. So, antigens of this type are described as thymus independent antigens. They don't need help or involvement of T cells. For simplicity, we will restrict the discussion at this point in the chapter to a thymus independent antigen. We won't be discussing about thymus dependent antigen. So suppose a person were to be exposed to Haemophilus influenzae type B, an encapsulated bacterium that can cause fetal meningitis. The capsule of these bacteria contains a polysaccharide that can bind to a tiny fraction of the body's B cells. So the B cells that bind the polysaccharide contain membrane-bound antibodies whose combining site allows them to interact specifically with that antigen. In this way, an antigen selects those lymphocytes that produce antibodies capable of interacting with that antigen. So, antigen binding activates the B cell, causing it to proliferate and form a population or clone of lymphocytes, all of which make the same antibody. Some of the activated cells differentiate into short-lived plasma cells that secrete large amount of antibody molecules. So unlike their B cell precursors, plasma cells possess an extensive rough ER characteristic of cells that are specialized for protein synthesis and secretion. Then immunologic memory provides long-term immunity. So not all B lymphocytes that are activated by antigen differentiate into antibody secreting plasma cells. Some remain in lymphoid tissues as a memory B cell that can respond rapidly at a later date if the antigen reappears in the body. Although plasma cells die off following removal of the antigenic stimulus, memory B cells may persist for a person's lifetime. It has been demonstrated, for example, that elderly individuals who were alive during the 1918 influenza pandemic still contain are generated by a process in which DNA segments are randomly combined. As a result, genes are invariably formed that encode antibodies that can react with the body's own tissues, which could produce widespread organ destruction and subsequent disease. It is obviously in the best interest of the body to prevent the production of such proteins, which are called autoantibodies. As they develop, many of the B cells capable of producing autoantibodies are either destroyed or rendered inactive. As a result, the body develops an immunologic tolerance towards itself. Then several principles of the clonal selection theory can be illustrated when we learn about vaccination. So Edward Jenner practiced medicine in the English countryside at a time when smallpox was one of the most prevalent and deadliest disease. Over the years, he noticed that the maids who tended the cows were typically spared the ravages of the disease. Jenner concluded that milkmaids were somehow immune to smallpox because they were infected at an early age with cowpox, a harmless disease they contracted from their cows. So, cowpox produces blisters that resemble the pus-filled blisters of smallpox, but the cowpox blisters are localized and disappear, causing nothing more serious than a scar at the site of infection. In 1796, Jenner performed one of the most famous and risky medical experiments of all time. First, he infected an 8-year-old boy with cowpox and gave the boy time to recover. 6 weeks later, Jenner intentionally infected the boy with smallpox by injecting pus from a smallpox lesion directly under the body's skin. The boy showed no signs of the deadly disease. Within a few years, thousands of people had become immune to smallpox by intentionally infecting themselves with cowpox. This procedure was termed vaccination after "vacca", the Latin word for cow. So Jenner's experiments were successful because the immune response generated against the virus that caused cowpox happens to be effective against the closely related virus that caused smallpox. So most modern vaccinations contain attenuated pathogens, which the pathogens that are capable of stimulating immunity but have been genetically crippled so that they are unable to cause disease. So most vaccines currently in use are B cell vaccines, which are such as that employed to fight tetanus. So tetanus results from the infection by the anaerobic soil bacterium Clostridium tetani, which can enter the body through a puncture wound. As they grow, the bacteria produce a powerful neurotoxin that blocks transmission across inhibitory synapses on motor neurons, leading to sustained muscle contraction and anaphylaxis. Asphyxiation, that means oxygen deficiency. So, at 2 months of age, most infants are immunized against tetanus by inoculation with a modified and harmless version of the tetanus toxin called toxoid. The tetanus toxoid binds to the surface of B cells whose membrane bound antibody molecule have a complementary binding site. These B cells proliferate to form a clone of cells that produce antibodies capable of binding to the actual tetanus toxin. This initial response soon wanes, but the person is left with memory cells that respond rapidly if the person should happen to develop a C. tetani infection at a later date. Unlike those immunizations, immunity to the tetanus toxin does not last a lifetime, which is the reason that people are given a booster dose every 10 years or so. The booster shot contains a toxoid protein and stimulates the production of additional memory cells. What if a person receives a wound that has the potential to cause tetanus and they cannot remember even receiving a booster dose? In these cases, the person is likely to be given passive immunization consisting of antibodies that can bind to the tetanus toxin. Passive immunization is effective for only a short period of time and it does not protect the recipient against a subsequent infection. Next topic is about T lymphocytes, action and mechanism of action, which we will be learning later.